So our next speaker today is Dr. Christoph Weidemann. Christoph is an associate professor at the Department of Psychology at Swansea University in Wales, and he currently does research at Columbia University's Department of Biomedical Engineering. And today, Christoph is going to be talking about neural measures of subsequent memory reflect endogenous variability in cognitive function.
Thanks for this, and thanks for the organizers. It's a great conference. I'll be presenting joint work with Mike Kahana, and I'll start off with a fairly obvious observation, although the theme is quite different from the previous talk. And just the observation that when we measure memory in the lab, there's usually a number of distinct phases in such an experiment. So we usually have a study phase where we ask participants to study a list of items. In the experiment that I'll be presenting today, this will be a list of words. And then there might be some period where they have to retain that information. And then later on, there will be some type of memory test where they have to retrieve the information and show that they encoded something earlier. And in the experiment that I'll present today, this will be a free recall task where they just simply, people just simply have to recall as many items as they can remember from the study phase in any order that they like. And performance in that memory task is a joint function of the processes that are involved at each of these stages. So when somebody successfully recalls an item, that requires that the item was successfully encoded and stored, and then later successfully retrieved. And obviously, a failure to recall an item could reflect a failure at each of these processes. So it's often quite hard to specifically attribute changes in performance in these memory tasks to individual components such as an encoding process. And the way neuroscientists have tried to address this issue is by collecting ongoing brain activity during these tasks. So for example, we can measure brain activity during the encoding session and then analyze this activity as a function of subsequent performance in that later memory test. And so the idea being that any brain activity that distinguishes between later recall or failure to recall might be associated with the encoding processes that then later allow this item to be retrieved. But there's obviously a lot of different ways to measure this brain activity. And in this study, I'll be looking at EEG recordings, and this is just one example of what this setup might look like. And there's lots of ways to analyze this data. So from each electrode, we'll get a time series like this. And the way I'll be analyzing the data here is to decompose that signal into different frequencies. So this is a little cartoon of what this looks like. And we're looking at power at a range of different frequencies, from low frequency fluctuations to high frequency changes. And to just give you an overview of what the data might look like from such a encoding list, this is the beginning of a study list. We have just 3 items here. The words judge, star, and actor, and I've bolded these words, the word judge and actor here, to indicate that these words were later recalled, and star was studied but not later recalled. And the associated brain activity is just plotted below. Time is on the x-axis, frequency is on the y-axis, and the colors indicate power at that time point, at that frequency. And when you look at this, look at the neural activity this way, it's very hard to make sense of it and to see any consistent patterns. But by just aggregating across all items that were subsequently recalled or all items that were not subsequently recalled, we can get a sense of to what extent brain activity distinguishes between subsequent recall. And I've illustrated this here so that you can see that if you aggregate all unrecalled items, that activity looks quite different from the activity for all recalled items. And you can also see that this change is quite sustained as a function of time. So the vertical line here is the onset of the item, and you can see this, these changes are quite long lasting. So from now on, when I'll be showing you these kind of data, I'm going to be aggregating across time and just show you power in different frequencies without resolving the time axis here. So it's very tempting to conclude by looking at this that, well, maybe this is a neural signature of encoding processes that determine whether an item successfully encoded so that it can be later retrieved. But an issue with that interpretation is that we don't have any direct control of whether or not an individual item is later recalled or not recalled. And indeed, there's lots of external factors that correlate with recall success. And so they present significant confounds in this analysis. And one of those variables is serial positions. This is just where in the study list an item is presented. I'm showing you this here. So what I'm showing you here is for individual items. We have 24 items in each study list. The probability that that item will be later recalled is a function of where in the list it was presented. And so you can see that items that were presented early in the list are much more likely to be later recalled than items that are presented in the middle of the list. And so when we calculate the contrast between recalled and unrecalled items, the recalled items disproportionately come from early list positions, and the unrecalled items are more likely to come from later list positions. And we can plot brain activity now instead of as a function of recall. We can plot brain activity as a function of serial position. And when we do this, we can also see marked changes in brain activity as a function of serial position. Just to remind you what the brain activity looked like as a function of recall, I've copied this here from the previous slide, and you can see that brain activity early on in the list looks very much like the brain activity for recalled items, and brain activity for later items looks very much like that for unrecalled items. And indeed, we can do the same kind of serial position analysis by just looking only at recalled items and also by only looking at not recalled items. And you can see in each of those cases, serial position effects are evident in the brain activity. And so they might contribute to any subsequent memory contrasts that we're looking at. And of course, serial position is just one of many external factors that affect the likelihood of individual items to be recalled. So the goal here is to try to distinguish between the endogenous variability that's associated with encoding processes and effects of external factors such as serial position. So I'll start off with just showing you a multivariate way to calculate a subsequent memory effect. So what I'm doing here is I'm combining brain activity from all electrodes and across all frequencies to train a classifier, just a logistic regression model here, to predict the probability of recall for individual items. And then we can evaluate the performance of this classifier just by correlating the output of this model with the recall status of individual items. And so this is a bit of an unconventional way to evaluate the performance of a binary classifier. In fact, this is a point by serial correlation because recall status is a binary variable. But I'm doing this to be better able to compare this performance with the performance of the other models that I'll present shortly. And so across all the participants, we can then plot this correlation or the distribution of this correlation, and we can see that it's positive, and this is our subsequent, multivariate subsequent memory effect. And so now the next step is to try to calculate a corrected subsequent memory effect where we statistically remove the effects of external factors. And so here, instead of predicting the recall status of individual items based on brain activity, we now, as a first step, predict the recall status of individual items based on these external factors. And specifically, I'm considering recallability here of individual items, and I can talk to you later about how we estimate this. The serial position, list number and session number. So we have multiple sessions for each participant, and within each session there's multiple lists, and so there could be practice and interference effects, for example. So we throw all this into this model, and then we use brain activity to, rather than directly predict recall of individual items, we now predict the residual recall after statistically removing these external factors. And then we can proceed just as previously by correlating the output of this model with the residual, and then we get our corrected subsequent memory effect, which is slightly lower, but still quite substantial. And you can see that a large part of that subsequent memory effect remains even when we statistically remove the effects of these external factors. I'm going to skip over this part and move on to another way to look at this by now calculating list level subsequent memory effect. So instead of predicting the recall performance for individual items, we can also just look at recall performance for individual lists. That gets rid of some of the external factors just by aggregating across them. So every list has the same number of serial positions. So any changes in recall performance as a function of serial position will just get averaged out when we look at lists as a unit of analysis rather than items. And we can do the same thing now. We have a regression model here because we have the probability of recall across the entire list of items. And then we can correlate the output of that model with that probability of recall, and we get an uncorrected list level subsequent memory effect. And we can see that works quite well. We have a positive subsequent memory effect, but of course, there's still external factors that could contribute to that effect. And so I'm proceeding the same way that I did for the item level subsequent memory effect. We're accounting for remaining external factors by first predicting the list level recall performance on the basis of these factors. So looking at the average recallability for items, for all the items within the list and list number and session number, and then as a second step, predict that the residuals of that model based on the brain activity. And then again, we can correlate the output of that model with those residuals, and we get the corrected subsequent memory effect. And you can see again, just as for the item level subsequent memory effect, that that corrected subsequent memory effect is only slightly smaller and very robust. So just to wrap up, we can conclude that the substantial item and list level subsequent memory effect, even after accounting for external factors. We have relatively small effects of the external factors on the SME, even though we can see that these external factors can predict recall performance quite well. They don't seem to be a major confound in the contrast of neural activity as a function of subsequent memory. And therefore, we can conclude that the subsequent memory effect largely reflect endogenous variability that's associated with cognitive function rather than effects of these external variables. So the paper just very recently got accepted at the Journal of Experimental Psychology, Learning, Memory, and Cognition, and the preprint is available on my website at cogsci.info. Thanks very much.
Thank you, Christoph. That was a fantastic talk. So I was wondering if you had like a kind of a prior manipulation or some words that were repeated more than others, would you expect the same result, or would you expect it to have an impact on the EEG?
Well, interesting. So, well, you could think of that as another external factor, right? If you repeat an item more than once, you essentially you could think of that as changing that item's recallability. So you're making some items more memorable in a way, and so you could model that the same way. I mean, it gets a bit tricky because you kind of it raises the question: are you only looking at how are you looking at the different the different encoding episodes, right? You could imagine that if you don't know that an item is going to be repeated, the brain activity at the initial exposure shouldn't distinguish between repeated and non-repeated items. But when an item is repeated, there's more going on than just the regular encoding process. You also recognize that the item was there previously. So I haven't done anything along those lines. I think you could probably model those effects in a similar fashion as to what I've presented here, but I think you'd have to think carefully about how to deal with those repetitions and the fact that that at the same time you're encoding that item, you're also retrieving the fact that you've seen it earlier in that same list.
Sorry, thank you.
Yeah. Okay. Well, thanks very much. I'm going to stop sharing. So,
okay.